Okay, next we'll be looking at how translation is initiated, and the initiation process is a bit different between the pro and eukaryote, so we'll look at them one by one. For the prokaryote, let's look at the players involved, right? We have the small and large subunit, right? The 50S large subunit and the 30S small subunit. There is an initiator tRNA, which refers to like the first tRNA to enter the ribosome, right? And it carries a modified amino acid known as formylmethylene, right? So the normal amino acid is methylene, a formal group is added to this amino acid, okay, for the initiator tRNA. And of course the mRNA with the Shine-Delgado sequence, right, specified by SD. So it's not a nucleotide sequence SD, right? There's no S and D in the nucleotide. Yeah? It's Shine-Delgado sequence. Alright, so the first thing that happened is that the mRNA would bind to the small subunit at the mRNA binding site via the Shine-Delgado sequence, right? And this binding is through complementary base pairing. So we call that the ribosome subunits, right, are made of a complex of rRNA and ribosomal proteins, right? So for the small subunit, at the mRNA binding site, there would be rRNA molecule that would have a complementary sequence to the Shine-Delgado sequence, right? So this binding to the mRNA binding site is via complementary base pairing between the Shine-Delgado sequence and the rRNA at the mRNA binding site. After that, right, the start codon would be nicely placed at where the future P site will be. Right, so the P site is found on the large subunit, which will combine later, right? So you will see that once the large subunit binds, right, it will just nice, the P site will just nice be at the AUG start codon. Right, so the next thing that happened before the large subunit bind is the binding of the initiator tRNA that carries the formylmethylene, right? So the anticodon of the initiator tRNA would be complementary to the start codon. Okay, lastly, the binding of the large subunit, and as mentioned, right, just nice the P site in the center here would just nice fit and where the AUG is and where the initiator tRNA is. Right, so now the translation initiation complex has been formed. Translation has been initiated. Right, the next tRNA, right, or rather the next amino acyl tRNA carrying the next amino acid to be added would then be able to bind to the A site, which we will look at later during the elongation process. All right. So that's the initiation for prokaryotes. Right? For the eukaryote, is slightly different. Okay, there is also the large and small subunit, 60S large subunit and 40S small subunit. There are proteins known as initiation factors. There's also an initiator tRNA, but it does not carry a formylmethylene. It just carries the normal methylene amino acid. Right? And an mRNA, right, which has a 5' UTR for binding to the small subunit. Okay, and of course, post-transcriptional modification has happened, so there's a cap, and of course, there's also the poly-A tail on the other side. Right, so the first thing that happened is that the initiator tRNA actually binds to the small subunit first, right, and that's stabilized by the initiation factors. So this is different, right, from the prokaryote, which the mRNA binds to the small subunit first, right? So for the eukaryote, it is actually the initiator tRNA that binds to the small subunit first. After that, right, this complex would bind to the mRNA via the mRNA binding site of the small subunit, which binds to the 5' UTR on the mRNA, right? So this binding again is due to complementary base pairing between the rRNA at the mRNA binding site and the 5' UTR. Okay, once that is done, right, the complex of small subunit with the initiator tRNA would move move downstream of the mRNA until the anticodon finds the start codon. Right, so let me show this again. Right, it moves downstream until the anticodon, if you can see my cursor, right, until the anticodon finds the complementary start codon AUG. Right, once that is found, right, then the large subunit would combine and displace the initiation factors. Right, then the translation initiation complex is formed, and translation is initiated. All right, so the process slightly different, right, from the prokaryote. But either way, right, the translation initiation complex is formed, translation is initiated, and it will be ready for the next stage of elongation.